Okay, here we have our host cell, which unfortunately is about to be invaded by the human immunodeficiency virus, which we have right here. And to describe the virus itself, it is surrounded by these glycoproteins, and what these glycoproteins help it do is bind to specific receptors on the host cell. Now, inside of it, what we have are two identical single strands of RNA and two molecules of reverse transcriptase. The RNA is represented by the red, and then these two dots, the green, is the reverse transcriptase. Now, what's going to happen is that the virus is going to attach to the host cell and their plasma membranes are going to fuse. Now, when they fuse, what happens is that the contents of the virus are released into the host cell. As you see here, the RNA and the reverse transcriptase, the green dots, are going to be released into the host cell. Now, once this happens, what we're going to have is this infected host cell. So what we have up here are the two strands of RNA, and then we have these green dots again, which are the reverse transcriptase. So what's going to happen is that this reverse transcriptase is going to catalyze the synthesis of DNA strands, which are complementary to the viral RNA. In the end, we have this double-stranded DNA that again is complementary to this RNA that came from the virus. Now, the next step is very important, and it is when the provirus is formed. This is our nucleus right here. Now, the way the provirus is formed is that this viral DNA that we just formed is going to integrate itself into the host cell's chromosomal DNA, which is represented here by this sort of tan yellow color, and then this blue is the viral DNA. So it incorporates itself, and this is very unique to what is called a retrovirus, which is what the HIV virus is. And it's unique because when this happens, when it integrates itself into the host cell's chromosomal DNA, in this way, it remains a permanent resident of the cell. Now, when this happens, what's going to happen is we're going to form more of this RNA, which is right here in the red. So the proviral genes, again, they transcribe into RNA molecules, which we have here. We have these two RNA molecules, which will serve as genomes for the next viral generation and as mRNAs for translation into viral proteins. And these viral proteins, again, will include this reverse transcriptase, the enzyme. We have the capsid proteins, and again, those glycoproteins that surrounded the original virus. So that's what's going to form from what we have here inside of the nucleus. So the next step now is we have this vesicle. And what this vesicle has are the glycoproteins. And this vesicle is going to go to the plasma membrane, and what's going to happen is it's going to pretty much release these glycoproteins and transport them to the cell's plasma membrane. And what we're going to have is something like this. So we have these glycoproteins that are on here on the host cell. And again, we formed, remember, these little blue dots right here are the capsids. This is the RNA, and these are the reverse transcriptase. And again, the vesicle transported these glycoproteins to the plasma membrane. Now what's going to happen is that these capsids, these proteins, are going to surround around the viral information. So the capsids are going to assemble themselves around the viral genomes and the reverse transcriptase molecules. And it's going to look something like this. So it's going to surround itself. They're going to surround this information right here. Then this is going to go to the plasma membrane of the host cell. And eventually what's going to happen is it's going to bud off. So it's going to bud off of the plasma membrane, And again, we had those glycoproteins that were attached here. And what's going to happen is all of that is going to bud off, and we're going to have this new HIV virus formed, which is going to proceed to invade more host cells. Thanks for watching.